Good evening, friends, and I welcome you back to the channel this evening. Today, we will discuss bacteriophages, and this is now the second part of our two-part series where we will discuss bacteriophages. Now, like we don't discuss before, bacteriophages now viruses them be, but they be special viruses because they affect or they invade bacteria. They will enter into bacteria, begin to perform different things from inside the bacteria. So now, why would they give them special name as bacteriophages? Meaning, say they fit chop or they fit colonize bacteria. Now, this one dey important for us to know. Now, the important thing we want to start our discussion with now say e get two phases or cycles where our bacteriophages they enter. The first one na lytic phase or lytic cycle, and the second one na lysogenic phase or cycle. Now, wetin this one mean? The lytic phase means say bacteriophages wey we don talk say they be viruses, many a times when they been find their way into a bacteria, many of the times wetin dem do be say the genetic component of that bacteria, they go take am up, come use am to cultivate or make many copies of themselves. Now, if they don do this many copies of themselves, they go create protein coats around themselves. Once they don do all of this one, wetin go happen na say that bacteria go come get many bacteriophages inside the bacteria. Now, no be say they go just dey live for inside the bacteria, they will come burst that bacteria, and then they will come out into the environment, and the bacteriophages will come plenty for inside the environment. Now, this process wey be say bacteriophages go plenty inside the bacteria, they go use up all the things wey the bacteria get in protein, in carbohydrate, everything wey e get, they will come burst that bacteria. Now, then we dey refer to as lytic phase. Now, this method we dey refer to as the lytic phase or lytic cycle. Now, this na wetin some bacteriophages dey do. Other bacteriophages no dey function like that. Wetin they go do na say if they don enter that bacteria, they will come take their own DNA or RNA, come incorporate am, join am into the DNA or RNA of the bacteria. That their viral genome go come dey live within that bacteria for many years. They no go cause lysis, they no go cause any problem. So those kind of bacteriophages, we dey refer to them as prophages. And wetin they do or wetin they undergo na the lysogenic phase or lysogenic cycle. Now, many a times, some of these bacteriophages, for some reasons, e get something wey we dey call induction. Something fit happen and they will come comot from that lysogenic phase, enter the lytic phase, meaning say they will comot from inside the genome of the bacteria, come begin dey replicate within the bacteria, come still burst that bacteria, meaning say they don move from the lysogenic phase or stage to the lytic phase. So this na two phases wey bacteria fit wey bacteriophages fit dey. They fit be dey for the lytic phase or they go dey for the lysogenic phase. Now, apart from that, another question we go ask na say wetin be the application of these bacteriophages? Wetin we dey use them for? Number one na say these bacteriophages, we don dey look at them as alternative for our antibiotics. And how this one go work? We don notice say phenomenon wey they call antibiotics resistance don dey on the increase. And wetin this one mean na say many of these antibiotics wey we get, they no dey effective again. They no dey work against this our bacteria. So this one come make scientists come dey look say, okay, we fit dey use these bacteriophages since say they enter into bacteria, the viruses wey they enter into the bacteria and destroy the bacteria, we fit dey use them instead of to dey use antibiotics. So we don dey look at them as alternatives wey we fit use instead of the antibiotics. Another important thing wey we dey use bacteriophages for na say we dey use them study wetin we dey call genetic engineering. We dey also use them study gene expression. When we talk genetic engineering, we dey talk about all the processes and ways wey we fit use manipulate genes wey dey inside organism. We go manipulate and come produce a better organism. We also get wetin we dey call apart from genetic engineering, we get wetin we dey call gene expression. And gene expression bin dey discuss the function of a particular gene. So we dey use bacteriophages study all these aspects for inside molecular biology. We also notice say bacteriophages dey help to shape the bacterial population. Because say they destroy bacteria a lot, they dey help shape bacterial population, they dey help shape evolution of bacteria. All these things na wetin bacteriophages dey do. We also dey use bacteriophages a lot in science. We dey study them a lot. Now, e get two major bacteriophages wey dey popular. One na T2 bacteriophage, the other na T4 bacteriophage. They dey very popular. And the T2 and T4 bacteriophage, they be member of the family Myoviridae. They be member of that family. Their shape na the icosahedral shape, and both of them they be double-stranded DNA viruses. So they're very important to know about them. Now, apart from that, we also know say the T2 bacteriophage, they favor the lytic cycle. Meanwhile, the T4 bacteriophage, they favor the lysogenic cycle. These are some of the small things we will discuss about the bacteriophage. Thank you for listening to this our lecture. Like our content, share our content, and have a wonderful day.